Okay, we're going to look at your Chapter 10 Cambridge review. This review is for Cambridge. If you're not in the Cambridge class, you're looking for the other video. This is for the Cambridge class. All right, let's dive right in. Number one, what are some advantages of asexual reproduction and sexual reproduction? Well, for asexual reproduction, it reproduces at a faster rate and makes an exact copy of itself. No other organism is necessary. Now, when we say it reproduces at a faster rate, we're talking about the cells themselves. Sexual reproduction is not identical. There are new genes or a mixture of genes, since half the chromosomes come from one parent and the other half from the other parent. So those are the advantages of asexual and sexual reproduction. And we'll talk a little more about that later, that you can add to number one. Number two, why is DNA bundled into separate chromosomes? Why is it beneficial? Well, this ensures that each daughter cell will get the same number of genes. The cell makes a copy of its DNA. Now, this doubles the amount of chromosomes, and as the cell splits, each new daughter cell gets the same number of chromosomes and the same number of genes. We talked about in class, when a human cell DNA duplicates and the chromosomes duplicate, it goes from 46 to 92. Well, if I have a cell that has 92 chromosomes and it splits, what's half of 92? 46. So each new daughter cell would wind up with 46 chromosomes in a human cell. It's different for every organism. For humans, it's 46 chromosomes. All right, moving along. Number three, when during the cell cycle are chromosomes visible? Well, they are not visible during interphase, because we're talking about the cell cycle here, and that is interphase, which is G1S, G2, the M phase, which is mitosis, and cytokinesis. And we're looking to see when they are visible. Well, they're not visible during interphase. The only time they are visible is during cell division, which is the last part of the cell cycle, during mitosis. Now, prophase is when they first become visible, but it's not asking us when they first become visible. It's asking us when they are visible. But you can also see chromosomes during metaphase, meta meaning middle. We can see the chromosomes lining up in the middle, and because we can see them line up in the middle, we know they're visible also during metaphase. We also know they're visible during anaphase, because the spindle fibers come out of the centrioles, attach to the chromosomes, and pull the duplicated chromosomes apart. So they are visible during prophase, metaphase, and anaphase, and that is part of cell division, which is the last part of the cell cycle, which is mitosis. All right, number four, know the phases of PMAT. And when you hear PMAT, you should think mitosis right away, as well as interphase. Know what happens during each, including when DNA becomes replicated. All right, this is a major part of your test. It is a major slide, a lot of information, but we spent an entire class period really diving deep into this, so you should be pretty familiar with it by now. So interphase consists of G1, which stands for gap. Now, that's incorrect phrasing, because it's not a gap of activity. The cell does most of its growing in the G1 phase. It does not stand for growth, it stands for gap. The second part of interphase is the S phase, which is known as synthesis. The DNA becomes replicated during the S phase, and centrioles begin duplicating. And the centrioles are where spindle fibers come from, attached to duplicated chromosomes and get pulled apart. G2 is both centrioles are finally formed and will be used to pull the duplicated chromosomes apart during anaphase. PMAT, prophase, the chromosomes become visible and the nuclear membrane disappears. Metaphase means middle. The chromosomes line up across the middle. Anaphase, the chromosomes are pulled apart by spindle fibers, which come from the centrioles. And telophase, the cytoplasm is getting ready to split, and that will actually happen during cytokinesis, forming two daughter cells. So go ahead, pause here. Make sure you have all of this. Take a look over it. And if you have any questions, please text me, because there's a lot going on here. Okay, number five, during cell division, what do animal cells have? What do plant cells have? Okay, so animal cells have what's called a cleavage furrow, and that is right here. You can see it's pinching inward, and that's because the cell membrane is very flexible. If you look at a plant cell, the cell walls are not very flexible at all, and we call this a cell plate. So plant cells have a cell plate, and then animal cells have cleavage furrow. Right after this happens, whether it's a cell plate forming or the cleavage furrow happening, once the cell actually splits, what will happen is interphase will begin once again. All right, number 6, why do cells eventually stop dividing? Well, this is a very simple answer. That when they come in contact with other cells, they will stop dividing. For example, when you have a cut on your skin and your new skin cells are going to cover that cut up, they will stop dividing when they come in contact with other skin cells. You may be asking, well, why do I have dead skin cells? How are those reproduced? When you have dead skin cells, they flake off and your body will form new skin cells to replace the existing dead skin cells. All right, moving along, number 7. Cancer is a serious disease. What type of organisms are affected by this disease? Well, we have both multicellular organisms, which are large complex organisms such as us. We also have unicellular organisms, which are just made of one cell. We're looking to see which ones are affected. And cancer affects multicellular organisms. Now, some of the cells in multicellular organisms continue to grow out of control and create tumors. And this really is the dead giveaway when you start thinking, I don't want to say common sense, but when you start really thinking about it, if an organism is made of one cell, there's no cells that it has inside of its body to grow if it's only made of one cell. Organisms that are multicellular have many cells in their body and they can grow out of control, as opposed to a unicellular organism, which is just made of one cell. It has no extra cells to grow. Number 8, cells become specialized and turn into something specific. What is this process called? You can do all of this in one word. We did this when we started talking about the vocab, and that is differentiation. That is when the cell becomes specialized and it's going to turn into whatever it's going to turn into. Whether that is a lung, heart, whatever. Now, there's different types of stem cells that do differentiation, and we're going to talk about that on this question. So stem cells have the potential to turn into different types of cells. There are totipotent stem cells, which can turn into literally anything that the body needs. Pluripotent stem cells that can turn into almost anything. And then there's also multipotent, which means stem cells that can turn into cells from the organ in which they came, adult stem cells. So for example, if I take adult stem cells from the brain, they can only turn into cells that have to do with the brain, such as neurons. If I take stem cells from the heart, I can only turn them into cells that have to do with the heart. But if you have a heart attack, you're not going to take stem cells from the heart because the heart would be damaged. All right, number 10, think critically about what stem cells can do and provide for us in the future. For example, it can heal damage done by a heart attack. They're going to give you a list of scenarios you would have to or choose which one would benefit from using stem cells. Heal damage done by a heart attack would be an example there. All right, number 11, cells give off carbon dioxide as a waste product. What is a factor in how much waste it produces? Well, the volume of the cell is the factor. Volume is how much space the cell takes up. It says it takes up, and we are talking about the cell. Volume is how much space the cell takes up, and the larger the cell, the more waste it will produce because it's going to have higher demanding taxes on the DNA in forms of energy. It's going to need more energy, and because it's going to need more energy, it's going to produce more waste. Number 12, what is apoptosis and what is an example? Apoptosis is programmed cell death. These cells are programmed to die. An example would be cells between toes and fingers, and this is why you have spaces between your toes and fingers. You can also see this done in the mice between their toes. There were cells during development, apoptosis set in, and the cells between the toes died, just like what you see in humans. All right, number 13, where can you get stem cells from that is not controversial? You can get stem cells from adult cells instead of an embryo. It is important to note that these stem cells do not act the same as embryonic stem cells. The adult stem cells are only multipotent, and we talked about the difference between totipotent, pluripotent, and multipotent. And you can get stem cells from adult cells. A lot of people who are against stem cell research will say, oh, you can get them from the adult. Yeah, but they won't turn into whatever you want, and you have to decide where your principles and beliefs are to decide if you're for or against stem cell research, but that's for another time. Moving on to continuing to review. So what are some problems that happen when cells grow? There's a multitude of problems that happen. These are the two most common. Now, as cells become larger, more demands are placed on the DNA DNA, and we just talked about that a couple questions ago. And the cell has more trouble moving waste and nutrients across the cell membrane because they are much larger. More demands are placed on the DNA. That is one whole thing. The cell has more trouble moving waste and nutrients across the cell membrane. That is the second thing. So if you're asking where the two are, they're right here in that sentence. All right, moving on. What type of environment would be best suited for asexual organisms, and what about sexual organisms? So this one, they're looking for something very specific. Now, asexual organisms would thrive in an environment where environmental conditions are stable. And if you think about it, this really makes sense. Remember, asexual organisms make an exact copy of themselves. The cells being made would be exact copies of the parent cells. And think about it like this. If the parent cells can survive the conditions, then so can the daughter cells. If there's a change in the environment, the cells may not survive since there's no changing of the DNA. The DNA would be the same. Well, if the parent cells can survive and we're making an exact copy, so would the daughter cells. I mean, that makes perfect sense. Now, sexual organisms could survive a change in the environment since there would be a mixing of genes, since two organisms are needed for passing on half of their chromosomes. For example, half your chromosomes in your body, since you have 46, half of them came from your mom, 23. The other 23, or the other half, came from your dad. And there'd be a mixing of genes. All right, number 16, and that's just what we wrote right there. Number 16, what do the levels of cyclin in a cell do? In general, it helps the cell pass on to the next phase of the cell cycle. If there's no cyclin, the cell would continue to grow and continue replicating DNA, resulting in an overload of DNA. And too much DNA makes the cell inefficient in exchanging materials. Since there's twice as much DNA as there was to begin with, this makes less room in the cell. All right, number 17. How are cancerous cells different from regular cells that are considered non-cancerous? Well, non-cancerous cells grow in a controlled way. Their cyclin works like it should. Cancerous cells grow out of control, and when they start growing out of control and you have a lump of cells that are growing out of control, it's going to form tumors. Now, if the cells go on untreated, these cancerous cells, they can spread to the rest of the body, causing serious medical issues and possibly death. And think about it like this. If a cancer cell reproduces through mitosis or asexual reproduction, it's going to make an exact copy of itself. So if a cancer cell reproduces, it's going to make a copy of the cell that's not functioning like it should, and it's just going to keep replicating. If you don't get rid of those damaged cells, cancer is going to continue to spread, and when cells are not working like they should, this can result in serious medical issues and possibly even death. And that brings us to number 18. What is the P53 gene and what effect does it have on cells? Well, the P53 gene helps regulate how fast cells divide. It helps also control cell growth and the regulation of cells dividing. So that helps with preventing cancer. And if you have cancer, it's a possibility the P53 gene is not working like it should. All right, number 19, someone who has a mass of cancer cells can be treated by, and there's a couple different ways you can treat cancer. We're going to talk about the two most common forms. One that everybody's pretty familiar with is chemotherapy, and that would damage all the cells it's being targeted to. This includes the healthy cells. Now, the cells that are cancerous would die off. Now, the healthy normal cells, they would still receive damage from the chemotherapy, but because they're healthy and normal, they would be able to recover and heal and bounce back. So the damaged cancerous cells would die off. The healthy cells, they would bounce back and recover because they would be damaged by the chemotherapy as well. Another one that a lot of people forget is actually physically removing the tumor from surgery. Removing it through surgery from the body. If you take the tumor out, you're taking out the cells that grow out of control, and they won't be able to replicate because they've been taken out of the body. Now, this has to be done before cancer spreads. If cancer starts spreading, you're not going to be able to take out all the tumors or all the cancer cells in the body, and you're going to have to resort to chemotherapy. Number 20, how does the ratio of surface area to volume impact cell growth? Well, the volume increases faster than the surface area, which causes the ratio to become much smaller, the ratio of surface area to volume. Moving materials across cell membrane become more difficult, just like we mentioned earlier. Now, a normal cell would divide into two daughter cells before it gets too large. If we have a cell that's not functioning like it should, we're looking moving materials across cell membrane become more difficult. But if it's a normal cell, before it gets to that point, it'll split into two daughter cells, an exact replica of the original cell. All right, moving right along, almost done. Number 21, how does mitosis and cytokinesis relate? What happens if one doesn't happen? In the past, a lot of people only look at this first part of the question, not the second part. We're going to do the whole thing right now. Now, mitosis is the division of the nucleus. Cytokinesis is the division of the cytoplasm. Remember, we said mitosis and cytokinesis, they happen at the same time. Towards the end of mitosis, cytokinesis begins happening. And in mitosis, we're taking those duplicated chromosomes and we're putting them into two separate nuclear membranes. That way, when the cell splits, each cell has a nucleus. Now, if there is no cytokinesis at all, the cell would contain two nuclei and twice as much DNA. Because remember, the DNA and chromosomes replicate. So you're going to have one cell with two nuclei instead of one nucleus and twice as much DNA. That's not going to benefit any cells at all, because the whole point of replicating DNA and chromosomes is so the cells can continue to divide and the organism can grow. Now, if there's no mitosis, then each daughter cell would roughly only receive half the chromosomes it should have, since there was no replication of the DNA. You would have a cell that would have 46. If there's no replication, then when it splits, it would have 23. The daughter cells would have 23 chromosomes and 23 chromosomes in the other daughter cell. It's not going to have all the information that it needs. All right, number 22. Why is stem cell research controversial? It's extremely controversial, and all you have to know for this is where stem cells come from. And stem cells that are totipotent or pluripotent are taken from embryos. An embryo is a unborn child, a fetus. That is what science says. You can say that it's unborn human baby. You can say whatever you want. It depends on your principles and beliefs. We're just sticking with what science says here. Now, when the stem cells are taken, the embryos are destroyed, since there are no stem cells to eventually turn into what the organism would need for survival. When you take the stem cells, there's no more development for that fetus, the unborn baby, whatever you want to call it. It's up to you. But once you take those stem cells out, the embryo is destroyed. It will be no more, and it's not going to be able to grow. It will die. Number 23, the last part, something we've already done, but it's a good review. Know the main events of the cell cycle, including interphase, G1, S, G2, and M phase. Interphase consists of G1, S, G2. Just a review. G1, remember, it stands for gap, but that is an inaccurate phrase, because the cell is going to do most of its growing in the G1 phase. Then it brings you to synthesis. The DNA becomes replicated. Centrioles begin duplicating. They finish duplicating in G2. And remember, centrioles are where the spindle fibers come from to pull the chromosomes apart during anaphase. M phase is mitosis, and when you hear the M phase or the word mitosis, immediately you should be thinking of PMAT, which is prophase, metaphase, anaphase, and telophase. Something that is missing is cytokinesis, which happens at the same time as mitosis, usually during the late anaphase or early telophase stage. It does not happen after mitosis, but during mitosis. That's going to cover everything for the Cambridge test for Chapter 10. Please study hard, because this is one of the more difficult tests that you'll have this year. If you have any questions, please let me know. Good luck, and I'll see you on test day so you can earn that A.